Hello, everyone. My name is Marissa Malchione, and I am a senior manager with the Influenza Vaccine Innovation Team at Sabin. And I am so pleased to welcome everyone to the second installment of the Flu Vaccine Forum. This is a webinar and podcast series that's hosted by the Sabin Vaccine Institute, as well as the Influenza Vaccine Roadmap Initiative and the Global Flu Funders Consortium. So in today's discussion, our excellent lineup of panelists is going to unpack the challenged pathway that promising next-gen and broadly protective influenza vaccines face on their journey to the marketplace. And throughout the conversation, we'll be highlighting the influenza vaccine roadmap, the IVR, and its key milestones that can help to navigate the barriers that lie ahead. So before we get started, let me introduce our panelists. I will start first with our moderator for today's discussion, Dr. Diane Post, who is Chief of the Viral Respiratory Diseases Section at NIAID. We also have with us Dr. Norman Baylor, who is a trustee of the Sabin Vaccine Institute and former director of FDA's Office of Vaccines Research and Review. Dr. Scott Hensley, who we are excited to have profiling his lab's multivalent mRNA flu vaccine candidate, and he is a professor of microbiology at University of Penn's Perelman School of Medicine. And then Dr. Amin Khan, who is Chief Science Advisor of the mRNA Hub program at Afrigen Biologics and Vaccines. So a big welcome to the panelists. Thank you for joining us. And before we get started, I just want to let those tuning in know that we will be doing an audience Q&A towards the end of the webinar. So please feel free to send your questions in the chat at any point during the discussion, and we will answer as many as we can. And with that, I will hand it over to our moderator, Diane Post, to kick off the discussion. Diane?
Wonderful. Thank you so much, Marissa. Thank you so much for having us today. It's really exciting to be here to speak with our expert panel and to discuss such an important topic. So for the discussion today, we're going to start by having Dr. Scott Hensley discuss his game-changing influenza vaccine candidate. And then we will move into our panel discussion where we'll talk about regulatory challenges, establishing a pathway for approval and deployment of next-generation vaccines for influenza, and then we'll touch on regulatory partnership with industry. So it's my pleasure to have Dr. Scott Hensley now pull up his slides, if that's okay, Scott. And if you could briefly profile the UPenn vaccine candidate, describe its current development status, and discuss future testing plans for your vaccine candidate.
All right. Well, thank you, Diane. It's a pleasure to be here today. Can you see the slides okay?
Yes, they look good.
All right, there we go. Okay, so as Diane just said, I'm a professor of microbiology at Penn and also the director of the Penn Center of Excellence for Influenza Research and Response. And today I have 4 or 5 slides just to introduce the audience to our new multivalent mRNA flu vaccine. I'd like to start with this slide from a review that Mike Warby, Stanley Plotkin, and myself wrote a few years ago. And what this slide shows is the importance of our initial exposures to influenza and how that shapes how we respond to flu viruses later in our life. So let me walk you through it. If we're infected with a red virus in this example as our initial infection, for flu, we get infected by the time we're 3 years of age. And then we go on to get infected with a blue virus. What we've learned is that we have very good immune memory against, in this example, a red virus. Now, if we get infected first with a blue virus as our very initial encounter with influenza, and then go on to get infected subsequently with a red virus, what we find is that we have very good immunity against that first virus, in this case, a blue virus. Now, Thomas Francis in 1960 first observed this, and he described this as original antigenic sin. And he made it clear in his essay from 1960 that the whole sin of this situation is that we are only exposed to a limited number of viruses early in our life, and therefore we develop very narrow immune memory that can only recognize a limited number of viruses. And in this classic essay that he wrote in 1960, he concluded by envisioning vaccines essentially that could replace original antigenic sin with what he described as antigenic blessings. And so that was our idea of when we started to develop this multivalent mRNA vaccine. Our goal was to develop a vaccine that would elicit a baseline level of immunity in infants, essentially, so that they would have immune memory to be able to be recalled later in life when confronted with the diverse pandemic or potential pandemic influenza strains that we encounter today. So you can see here in panel C, the idea is rather simple. Can we design a vaccine that makes our initial encounters with influenza a multivalent one that has epitopes from all known influenza strains? And so we began working with Drew Weissman as early as 2015 on these mRNA-based vaccines. And it's interesting, after we've published our study on the multivalent vaccine last year, many folks said, well, isn't it a good idea that we're taking the COVID technology, the mRNA technology from COVID, and applying it to flu? It's actually the other way around. Drew's been working on many different vaccines, and we've been working on the flu components of these vaccines as early as 2015. So can these mRNA vaccines provide this initial blessing of induced immunity using the exact language from Thomas Francis's 1960 essay? I think many viewers at this point know the story with mRNAs. These mRNAs are modified in a way that they are not sensed immediately by the innate immune system. So this nucleoside modification and the purification of the RNA is very critical. In the case of COVID vaccines, these vaccines express the spike protein of SARS-CoV-2. In the case of our influenza vaccines, we have hemagglutinin as the coding sequence. Hemagglutinin is the major neutralizing and antibody target for flu. And importantly, these mRNAs are encapsulated in something called an LNP, a lipid nanoparticle. And these lipid nanoparticles not only shuttle the RNA into our cells, but they provide important adjuvant properties as well. So conceptually, here is the 20HA mRNA vaccine. It's a rather simple idea. What we did was we created 20 individual mRNA LNPs, each encoding for a single hemagglutinin that represents the 18 different influenza A types and the 2 influenza B lineages shown in this slide. I couldn't give a talk without having at least one data slide, so here it is. And I love this experiment. This is a simple experiment where we immunized mice with 1 dose of this vaccine, and we measured antibody levels, so that's shown on the y-axis, using serum from these mice either 28 days or 118 days after vaccination. And here we quantified how well these antibodies could react to the 20 different immunogens in our vaccine. And I think you can appreciate from this slide that we were able to achieve strong antibody responses against all 20 components. No real immune biasing towards one component or the other. And when I first prepped for today, I had 20 additional slides with data, and I figured that's not really the point of today. Today is to explore some of the regulatory issues and whatnot related to advancing not only this vaccine, but other universal flu vaccines. So I summarized just a few key points on this slide here. This vaccine that we've been working on has been tested in mice, ferrets, as well as non-human primates at this point. And the vaccine prevents infection with matched viral strains. You might be asking yourself, what does matched and mismatched mean in this sense? There's 20 different influenza subtypes or lineages, but within each subtype, there's a zillion different strains. As many of you know, flu is constantly changing. There's a lot of variation within each subtype. So when we challenge animals with a challenge strain that's actually matched with the precise HA that is in our vaccine, we see complete protection. But when we challenge with something that's very much mismatched, we don't necessarily see prevention from infection, but we see prevention from severe disease and death. And this is what really mimics a pandemic situation. We don't think that we'll be able to predict what pandemic strain might circulate 20 years from now. But the idea is not necessarily to elicit antibodies to prevent infection of those strains, but rather to prime the immune system in a way that immune memory is set so that these memory B cells and T cells can be quickly recalled and adapt to the actual strain that emerges down the road. So this is a very challenging issue when we think about how to get this vaccine through regulatory processes. The goal is really to prevent severe disease and death. I will just say that this is sort of analogous to the situation that we have with our original SARS-CoV-2 vaccines and Omicron lineages of SARS-CoV-2. In that case, there's a major antigenic mismatch. So those original SARS-CoV-2 vaccines did not necessarily prevent infections with these Omicron lineages of viruses, but what we see study after study is that they continue to prevent and protect against severe disease and illness, and that's the goal of our vaccine. I will also point out that while our original goal was to elicit antibodies in animals and eventually including humans who had never been exposed to flu, what we found is that this vaccine also elicits strong antibody responses in animals who've already encountered flu. And that's good news for adults or anyone that's older than 3 years of age. We like to think of this as absolution of original antigenic sin. So the current status is this. We are producing GMP lots of this vaccine as we speak, and we plan on doing human phase 1 testing, hopefully as early as 2024. Okay, so those are my slides. I'd like to acknowledge Claudia, who was the grad student in my lab, who did most of the experiments for the paper that we just published on this. She is now working at Pfizer. And of course, Drew Weissman, who we've worked collaboratively with and who actually produced these vaccines. I'm going to stop sharing my screen if I can figure out how to do that and hand it back to Diane.
Thank you so much, Scott. It was really great. So a few questions for you about your vaccine. What potential role do you envision for your vaccine candidate? Are you thinking that this will eventually replace seasonal influenza vaccination, or is the focus really on serving as a pre-pandemic vaccine?
Yeah, so that's a great question, a question I get a lot. This is not a vaccine to replace seasonal vaccination because the vaccine does not necessarily elicit neutralizing antibodies against all of the different strains that we can encounter, even seasonal flu, as well as the thousands of individual pandemic flu strains that are out there. The goal of seasonal flu vaccines, and why we update these on such a regular basis, is to elicit neutralizing antibodies against viruses that are circulating. Now, most of the neutralizing antibodies target these variable parts of the hemagglutinin protein of flu, and so constant updates are essentially needed in order to achieve that neutralizing antibody response that prevents infection. We should not give up our pursuit of eliciting neutralizing antibodies against seasonal flu. That's important to continue to do that work. Our vaccine, on the other hand, is meant to elicit memory B cells and T cells. In the case of memory B cells, even when we get exposed to a very different virus, these memory B cells can undergo a process, as you know, Diane, called somatic hypermutation, which allows these B cells to then, that are already primed in this memory state, to be able to develop and hone in on this new antigen. We think if we have memory B cells against all 18 different influenza A subtypes and influenza B, that we'll have essentially the B cell repertoire that can be diverse enough to respond to any subtype as it emerges in the future.
Yeah, thank you, Scott. That's great. So you mentioned about updating the strains, in the regular seasonal vaccine process. Do you anticipate having to update the strains for this vaccine? And if so, how frequently? And would you do them all at the same time, or could you maybe update just an individual strain?
Yeah, we've thought about updating vaccine strains, and we certainly can consider that. But the goal, again, is not necessarily to choose the right strain. What is the pandemic flu strain of 2030? What is the pandemic flu strain of 2045? We don't know, right? We don't know when these pandemic flu strains will emerge, and we don't know even what subtype will cause our next pandemic. And so we can consider updating our vaccine strains to more closely match whatever seems to be the highest pandemic threat today. And in fact, I'll give you an example. The H5 component of our vaccine was based on an earlier H5 strain from 2004, 2005. And we're considering updating that right now to more closely match the H5 strains that are sweeping across the world in birds today. So we can consider doing this, but I think the overall goal is not necessarily to always be playing catch up to these viruses, but just establishing a certain level of immune memory against all subtypes, and not necessarily getting it right of the actual clade or viral strain that emerges, but just covering all of our bases on some baseline level of immunity.
So potentially priming with every type of HA is really exciting, and I think it would be a huge improvement in us in our vaccines. Can you talk a little bit more about what makes the mRNA platform so appealing specifically compared to the others that are out there?
Well, there's a number of things. So if we just think about production, okay, can we make 20 immunogens? Can we make 100 immunogens? Uh, if we think about doing this with recombinant protein or standardizing egg-derived antigens from this number of antigens, that's a big ask. For mRNAs, this is relatively simple. The RNAs are made, they are made in vitro, essentially, and it's very easy to update these vaccines and produce multivalent vaccines. This is all plasmid-based expression of RNAs. So that's one consideration. The other consideration are the properties of mRNA vaccines themselves, I think, contribute to the induction of many different types of immune responses simultaneously. This gets into the weeds a little bit for I'm sure some of the audience, but a lot of the action of where immune responses are primed are called the germinal centers, which are in our lymph nodes. And the germinal centers, you can think of this area where B cells and T cells meet up and get activated. That's where they come in contact with antigens. And mRNA vaccines, they elicit this very long-lived germinal center reaction. There's lots of opportunities of B cells and T cells of all different types to get activated following vaccination with these mRNA vaccines. With conventional vaccines, of course, we see some induction of germinal centers, but it's not as long-lived. And we think that long-lived germinal center reaction allows for this very broad antibody response that we see with our vaccine. So we're continuing to look at this at more of a basic mechanistic standpoint, but we think both the practicality of mRNA vaccines as well as some of the intrinsic properties of these vaccines make them a good candidate for delivering universal flu vaccine antigens.
Thank you, Scott. So one more vaccine-specific question. I know your group does a lot of research on the impacts of pre-existing immunity for various populations, children versus adults, and the potential consequences of the first exposure and how that's super important for affecting how we respond to other strains later in life. And I know you started with that today with your first slide, talking about antigenic sin. Could you maybe just touch on a little bit more the importance of this and why this vaccine is anticipated to not be impacted by previous exposures, and any implications of this vaccine being a first exposure for children?
Yeah, so it's a great question and something that I just think is fascinating. I was born in the late 70s, and those early events in the early 80s have impacted in my case, have impacted how my immune system responds to flu viruses today. We kind of leave it up to dumb luck, right? Our immunity to flu is based on our year of birth and the specific sets of viruses that just happen to be circulating in those formative years that we had when we were 1, 2, 3 years of age. It's remarkable that those early immune imprints affect us so many decades later. We don't completely understand the immunological basis of this, but we think that these initial childhood exposures are the best in inducing these memory B cell responses. And that's why our goal going into this was to develop a vaccine that could be given to children very early in their life, so that every B cell, whether it was an H1, an H2, H3, H4, that every B cell, subtype specific B cell, was sort of on equal footing, so that if, again, there wouldn't be an immunodominance against one subtype or the other. Now, it's just icing on the cake that we found that our vaccine, at least in animal models, not only elicits these balanced responses in naive animals, but we've seen that they elicit balanced responses in animals that have already seen flu. So for these experiments, we infected animals with H1N1 or H3N2 to mimic seasonal flu infections. And then we came back after that and then vaccinated, and we found that there wasn't just a focusing of immune responses on those initially infecting subtypes, but again, broad immunity. So we don't understand this. Why is this the case? We think it has something to do with my answer to your last question. Again, these long-lived germinal center reactions. When you have these long-lived germinal center reactions and a lot of antigen around, we're not so reliant on immune memory. there's enough antigen to go around. Even the naive B cells who haven't been primed yet have an opportunity to get stimulated in this situation. And so further studies are needed to try to understand this. Is this something unique about mRNAs? Does this have something to do with the adjuvant properties of the LNPs themselves? We don't know the answer yet. And these are the kind of mechanistic questions we're looking at in the lab right now.
That's great. Thank you so much, Scott. So we're going to move on to discussion with our full panel. I'd like to welcome Dr. Norman Baylor and Dr. Amin Khan. Thank you both so much for being here with us today. Universal influenza vaccines that can protect against multiple strains, like the candidate that Scott just told us about, it really could help to prevent the next pandemic. However, there are lots of hurdles after initial preclinical discovery of a novel vaccine candidate. These issues can include identifying appropriate biological standards, issues with process development and manufacturing, demonstrating efficacy, and defining correlates of protection. So a question for the whole panel: what do you see as the major challenges and key considerations that we should be thinking about and discussing when moving from the bench to the clinic? So maybe I can start with Amin.
That's an easy question.
Yeah, I know.
Actually, first, let me just say, Scott, beautiful work, really. And I mean, you published this in November. I think it was in Science, maybe. I can't remember exactly, but it's truly wonderful work. It's also, in many ways, groundbreaking in terms of the concept of the vaccines and the way that we look at them, or we have looked at them in the past. And you're proposing a new paradigm. And I think one of the biggest challenges here from a regulatory perspective, and here, of course, you know, we will all defer to Dr. Baylor, given your background and experience. But this sort of sticks with me, which is that this is a new vaccination paradigm, right? Whereby you want to vaccinate infants, young children, right? Not necessarily to protect against a disease in the next season, but something that may happen in the distant future, right? And the vaccine is not designed to elicit an immune response for the anticipated specific pathogen, right? And I think that is really fascinating, and that is likely to present more challenges, in my humble view, from a regulatory and development point of view, than the many hurdles that have to be overcome in terms of, you know, influenza surveillance, the production of the seed viruses and the distribution, et cetera, and the annual updates that we see today with seasonal vaccines. So I think that, in my view, is going to be a much bigger hurdle. How do we actually develop this for a product that is going to go into young children? And you know, I'm really looking forward to hearing Dr. Baylor's response to this question. I also have some ideas, speculative things about how you might actually navigate your way to ultimately giving a vaccine to young children.
That's great. Norman, I think you're on mute. We can't hear you.
How about now?
Yes, perfect.
Okay,
now.
Sorry. I want to echo Amin's comments also, Scott. I mean, it's great work. I'm glad we're still pushing flu, trying to, you know, better these vaccines. It's definitely well needed. But there are a lot of challenges with this approach. And so when I usually think about a product, a new vaccine, I'm thinking about what clinical benefit are we looking at? I'm also trying to figure out, you know, if you deploy a vaccine, what's the indication? I mean, the indication is really going to drive the pathway. And so when I think about a universal influenza vaccine, I mean, taking your vaccine, for instance, I mean, it does elicit a lot of questions. I mean, if I think about a strain change and in a regular seasonal influenza vaccine, we're not changing the hemagglutinin. necessarily. Most of the time we're not, but we're changing the strain. And we know that if there's a good match, then you get a pretty good effectiveness. You know, it's still not the best, and we can talk about, you know, is the hemagglutinin really the right target to get a vaccine that's of high efficacy? But if you take your vaccine, I mean, if we have the multiple hemagglutinins, and then how do you determine what strain you're going to include in those vaccines? Because the next season, the strain will potentially change, and it's not going to change for all the HAs. It's going to change for some. So I got to think about, how do I evaluate that in the context of an indication? Or do I have a simple, straightforward indication of this vaccine will reduce severe illness and death among by those vaccines, by those influenza viruses that are included in the vaccine? That would be a basic indication. And then I have to think about, now, how do I demonstrate that? Do I need to demonstrate that for each of those hemagglutinins? And that's a tall order in itself. So this is where the complexity comes in to develop the regulatory pathway. I mean, the two basic goals are simple. I want a vaccine that's safe, and I want a vaccine that's efficacious or effective. Getting to that point, that's the challenge. How do I demonstrate that with a vaccine such as this? And then it gets a little bit more complicated, because if we're thinking about, you know, the influenza is a global enterprise. So we can't just think about, we can't be just thinking about regional. It's a global enterprise. WHO wants a vaccine with a good breadth of protection, robust immunogenicity and safety. They want a vaccine that's durable. Their goal is the vaccine with 5-year durability. That's a tall order. And the vaccine has to be suitable for global use. So that really makes this, you know, I'm not saying it's impossible, because nothing's impossible. We have to figure out whatever product that comes forward to the regulatory authority, we have to have a way to evaluate that product. And it's just a challenge.
So, Norman, if I may ask you a question here. So might you envision that you actually start with using this approach for a seasonal vaccine, and then over the course of a number of years, you continue to add additional immunogens, right? Rather than switching them out. Ultimately, you may get to the same endpoint. It would take you longer from a development standpoint to have the vaccine profile, Scott, that you're describing here. I mean, is that a possible approach?
So when you say seasonal, so I take this multivalent vaccine and then deploy it against whatever circulating? Is that
yes, and then update it, but instead of switching out the immunogens on an annual basis, you actually continue to add to them.
The challenge for me is how determining the effectiveness, and you can do this. I mean, you can do it with an efficacy study, determining the effectiveness of this multivalent vaccine against what's circulating for a couple of seasons. I mean, like we do now. But again, how will you determine what strain you're going to use? If you could clarify that, Scott.
Yeah, I could comment on the strains a bit. So I've talked to evolutionary biologists and to try to refine this, and you know, so we can look at phylogenetic trees and we can think about what is the most ideal immunogen you would put in it. Do we just play catch up for H5? I gave that example. There's a clear H5 avian influenza, high path avian influenza that's circulating across the globe right now. Do we just kind of play catch up and constantly update it? And the idea is not necessarily that. If anything, you would want to try to find an immunogen that's sort of at the base of a tree, of a phylogenetic tree, and not just try to match everything that is circulating today. Again, the idea is not necessarily to match the strains, but rather to deliver immunogens against all of the subtypes, because there are conserved epitopes within subtypes. You can think of these 18 different influenza A subtypes and 2 influenza B lineages. There are some epitopes that are H5 specific that are essentially in every H5 strain. Those are the kind of B cells that we want to activate. And so I have some of the best minds in the world that are part of the center that I work with, and we've met and discussed this. And at the end of the day, I don't know if there's a simple answer about which strains to put in, but we don't think it matters from the goal that we are trying to achieve, which is just to have this fundamental level of baseline immunity. The other thing that you can think about, this is how it would play out. If we ever could get this vaccine approved, you can imagine the 20HA vaccine being delivered. And then in the case of a pandemic, now say the H5 that is circulating in birds jumps into humans and causes a pandemic. Well, then this vaccine would serve as a priming, and we would then get boosted. We would then make a monovalent mRNA vaccine in response to that pandemic. So when we think about it, I think the vaccine strain changes, I actually don't picture that many vaccine strain changes for this product. I think efficacy and showing efficacy, as you said, Norman, is probably the biggest hurdle. If our readout is not necessarily preventing infections, or if our goal is not just preventing infections, but preventing severe disease and illness, you don't want to have to wait for the next pandemic to know if this vaccine is working. And in that regard, I think we have to really develop correlates of protection to be able to examine if our vaccine elicits those types of immune responses that we know are associated with preventing severe disease and death. That is a hurdle where it is not just for our vaccine, but for any universal vaccine.
Correct.
That is not necessarily designed to elicit these, magical antibodies that will prevent all infections, because that's probably not going to happen. But if we can have the goal of just preventing people from dying of pandemic flu, we can't wait for that pandemic to happen in order to learn those lessons. We need to come up with other ways to think about effectiveness.
But I mean, when you say the baseline immunity, it begs the question, what's the level? I mean, again, thinking about an indication, thinking about the effectiveness, when I'm talking about reducing disease, reducing severe disease and death, I think that should be the target. I mean, I think that should be the target really maybe if we were looking at that directly for seasonal vaccines, we'd see a higher effectiveness. We probably would. But what is this baseline immunity? What level do I need for clinical benefit? That's where I'm struggling. What is that target? And I want that clinical benefit as high as I can get it. I mean, that's what we did for COVID. COVID, I mean, we didn't prevent infection, we didn't prevent transmission, but we did significantly reduce severe illness and death. And those trials bore that out.
Yeah.
And that is especially the case once we saw this major antigenic change in the form of Omicron. It was, you hear these words, sterilizing immunity, and but early on in the pandemic, before the Omicron wave, we really did see much closer to complete protection that prevented, measurable illness at all. But again, that is what I've been just amazed at. I support updating the COVID vaccines that we have today. But if you just look at those original mRNA vaccines based on the original SARS-CoV-2 sequence, even today, those vaccines 3 years later prevent against severe disease and death. And I think that is, so how do we get from there to learning more about flu? If we can understand the correlates of protection, for example, that are involved with that type of protection, maybe that starts informing where we have to start looking in the case of influenza. I really think it boils down to correlates of protection, because I think we can all agree that we can't settle on just having a vaccine that induces sterilizing immunity. against all influenza types. I think that's just too lofty of a goal at this point. And we can't wait for our next pandemic. So we have to find some bridging studies there. And I don't have the answer at all.
I agree with you 100% for the correlates. I mean, the correlate, if we had correlates for protection, that would make vaccinologists' life a whole lot easier. But that is a challenge in itself too, because if you think about, are we talking about the same correlates necessary for adults, children, and the type of vaccine as well? So there's a challenge in itself, but not to say that it's impossible. But yes, the correlates would make this a whole lot easier, if you will. But getting to those correlates, it's been a challenge. I mean, we don't. Go ahead, Amin. I'm sorry.
I mean, correlates of protection in the context that you're speaking of here. I mean, it's going to, maybe this is a naive viewpoint, it's going to require a large body of clinical data, right? So how do you get that? Right. So if I may ask, Scott, you said you anticipate starting your phase 1 clinical studies in 2024. How deeply have you looked at what your endpoints are going to be for that study? And I don't mean that the safety endpoints. I think those are probably obvious. But the other endpoints, your immunogenicity endpoints, so have you considered those? Because that sort of takes you on that journey, doesn't it?
Yeah, it's a good question. So the primary endpoints will be looking at immunogenicity as far as antibody titers, but we will do secondary analyses as well. In the case of prevention or protection against a pandemic strain, it may not actually be the soluble antibody that is present at the time of infection that is required for protection. Rather, it may be the cellular components, those B cells that can undergo adaptations now to start molding themselves against a new pandemic strain. So the primary endpoints of the phase 1 studies will be simple and obvious antibodies, which we can very quickly and reliably quantify, but we'll do some sort of exploratory analyses to look at cellular responses as well.
I mean, so there are no vaccines approved today that are based on cell-mediated immunity alone, right? So that's a huge hurdle in itself, right? Given the limitations of the analytical tools, et cetera. And in terms of the neutralizing antibody, will you measure neutralizing antibody titers? You said antibody titers.
Well, both, yeah. So antibody titers, binding antibodies, as well as neutralizing antibodies will both be measured. But the initial goal is to determine whether or not this vaccine can elicit antibodies against all of these diverse subtypes without this immune skewing towards seasonal strains that the human population has already been exposed to.
So I mean, yeah. So you can do you can take the sera and you can test against the panel of 20 or so viruses that you have, right? I mean, that's what you're planning on doing. I mean, that'll give you some good indications. Right? But the question, as it's been posed, is where do you go from there?
Yeah, that phase 2, phase 3 is that's the tough part. But did you see any interference in antibody response among the HAs in the animals?
No, we didn't see any interference, and in fact, what we were concerned with, animals that had first seen H1N1, now we give them a vaccine, in some cases, a year later. The concern is that they're going to have good immune memory against H1N1, and that our vaccine, since there's an H1 component in it, that it would just elicit so many H1 responses and fail to get the other 19 component immune responses started. We didn't see that at all. We saw induction of antibody responses against all 20 components in animals that had prior exposure. Here's the interesting thing. The levels of antibodies against those other 19 components were actually increased when we did these kind of experiments in non-human primates. Why could that be the case? Well, we think that infection, in this case with H1N1, probably primes very strong CD4 T cell responses that are actually cross-reactive. CD4 T cells actually recognize very conserved parts of viruses. so that when we come in with a single dose of this vaccine in pre-exposed non-human primates, you see better induction of antibody responses against all of the components, even the components that that animal had not seen previously, because there's already good CD4 T cell help. All of this is to say that these vaccines likely are not going to be biased towards these influenza types that we've seen in the past. And a lot of this might have to do with antigenic distance. So we have to look at this further. And of course, you know, H18 is much different compared to H1 and H2 and H3. And so studies are underway right now to try to understand that better with different prior exposures that either are close related or more distant related to the antigens that we have in the vaccine.
So you all have brought up a lot of the questions and challenges with the approval pathway of the vaccines, especially that have they target viruses with pandemic potential that have not emerged in the human population yet, right? So it's hard to test against these viruses that are not circulating in humans. So there's a lot of discussion about the use of human challenge trials to gauge effectiveness against multiple strains, including those that are not currently circulating. So just wondering your opinions about how important do you think those type of studies would be for demonstrating breadth and potentially moving towards licensure? And how likely do you think those studies, especially those using strains not currently circulating, are?
I'll start. I think the challenge studies could definitely be a contributor. They can definitely they will definitely help. I mean, I think the issue is developing the strains to the challenge strains, especially the challenge strains where we have not been in humans. I mean, we had this debate with COVID and challenge, and you know, no rescue, no vaccine, and the whole issue of safety. And I think that is something that, you know, will be a challenge, thinking about, you know, and also it could get a little perception wise hairy when you think about challenging people with the influenza virus that has never been in humans and the consequences of it, or the potential consequences of that. So that can get a little, you know, we definitely have that debate. But I think there is room for challenge studies to contribute here, but it's going to take a lot of discussion.
Norman, I agree with you, and I was party to some discussions a couple of years ago on this very specific topic outside of COVID and influenza and challenge in humans with non-circulating strains. It's a minefield, right? And the technical issues aside, you know, the fact that there may not be, you know, a route to rescue. I mean, the ethical considerations around this, but the other piece is the need, right? So if there is a compelling unmet need, then I think the discussion becomes a little easier. But we saw even in the context of COVID how difficult that discussion was, right? So having been around this block a few times myself with influenza, I personally was very, very reluctant to support human challenge studies with influenza strains that are not in current circulation. But having said that, I'm not an ethicist, and it will be an interesting debate, Scott, to go down that road.
And I would point out too that, again, the overall goal of eliciting immunity against very different strains, I'm sure in challenge studies, if we actually had, you know, approved challenge strains from H5 or H7, if it was matched to the vaccine, that's one thing. You expect very good protection. Again, if the readout is protection against severe disease and death, that is not something that you can recapitulate in a challenge study, obviously, right? So a challenge study, I'm not sure from this vaccine profile how much it advances. Of course, if you have a match of the vaccine strain and the challenge strain, you're going to see protection. But if the ultimate goal is to prevent severe disease, hospitalizations, this is not something that you can recapitulate in a challenge model.
But the challenge model, I mean, again, with all the complications, with a challenge model could give you a hint about correlates. So, I mean, in that sense, you know, again, those that are not circulating, I have some, you know, again, that's a potential safety issue. But some of the others, you know, that we have seen, H5, H7, I mean, it could potentially give you a hint of possible correlates.
But is a hint enough, right? So,
and that's the thing. It's a hint enough. But you build on, you know, I mean, you build on those hints.
Yeah.
Because I don't know how else you're going to. I guess the other hurdle for the challenge is with COVID, we're in the midst of disease, so the unmet need is very different. Here, the unmet need is out in the future. It could be tomorrow. It could be way out in the future. So you can't make the argument that this is an imminent threat as we did with COVID. And so with COVID, you could say, well, the challenge may be could cause harm, but we know the virus is going to cause harm. So, but we're not in that position with influenza unless we were in the midst of a pandemic. So getting, and again, I'm not an ethicist either, but getting an ethicist to say, well, yeah, we're preparing for way out in the future or tomorrow, we don't know. How risky is this?
Yeah, I mean, the other component is, Scott, you've acknowledged that, you know, the protection comes from also potentially from the CMI, right? And it's not just antibody mediated, right? And so for me, it's getting to those correlates is going to be tremendously challenging. I mean, I'm just looking at everything that's been done over the last 3 years on COVID. We have such comparison of different platforms for the vaccine, so much immunogenicity studies. I mean, it's been explored, yet we don't have a correlate for SARS-CoV-2 yet. Right? So I think that the hurdle is extremely high, I think, in terms of determining a correlative protection in this case.
So we're going to have to move on to the audience question and answers here. Before we do that, though, I'd like to hear from each of you how you feel the scientific community, regulators, and product developers can best collaborate to establish a pathway for these next generation flu vaccines as they move forward. So, Amin, what do you think? What is the best way to collaborate? And then any final thoughts before we move to the question?
Sorry, I was interrupting you. Look, I have a simple-minded view on this, right? It's going to take a coalition of regulators, of academics, of industry partners to come to the table and remove the barriers, right? And otherwise, you know, Scott, it's going to be a very, very long way.
And I would agree with Amin. These are really scientific barriers. They're not regulatory barriers. They're scientific barriers, and I think that it is going to take a coalition of everybody involved: the scientists, regulators, the World Health Organization. Because again, this is global. It's got to be done on a global level, and it really is going. It needs to have just a tremendous amount of discussion about the risk benefits, because I mean, from the regulators' perspective, the risk is that they're the risk assessors. How much risk are we willing to take? Because I mean, some of this we're going to have to move forward. We're going to have to reduce. We're going to have to take more risks. That's what I'm saying. And this coalition needs to define that risk. It can't be FDA has this level of risk. EMEA has this. We got to have the same. We got to agree upon the same level of risk that we take.
Yeah, I mean, this is a difficult problem, right? And everything we've talked about today is not just affect the vaccine that we're working on, but it affects other vaccines as well, right?
Yeah.
So we could say, heck with it, it's hard. Let's deal with it later. Yeah. And then after a pandemic happens, we can scramble. Or we can do something about it now. And it might not be a process that gets worked out in a year or 2 years, but what I'm hearing from today's panel is that we need to have a better idea immunologically of why some people are protected from flu, why some people get very sick and die. And I think we can all agree that we're not going to wait for the next pandemic to be able to, see if these, not just ours, but other flu vaccines are promising. And it's really identifying the human immune response and what's important to lead to protection by studying seasonal flu, probably. That's what's going to inform our vaccine. So it's hard, but I'm willing to jump into that, and I think it's a partnership. It's not just academic labs like myself, but industry and organizations like Sabin who need to get involved here.
And governments, because they have the checks.
Right.
Yeah. Okay, well, tied to what you were saying, Scott, a question has come in the chat, and the question is, how might multivalent mRNA influenza vaccines smooth the development path for innovative vaccines against other pandemic threats? So they mentioned universal coronavirus vaccines, Nipah virus vaccines. What are your thoughts on how this might help some of the other pathogens that pose a pandemic threat?
Yeah, it's a great question. I mean, there's different problems with each of the examples that you list. Certainly, the COVID pandemic, I made a point to say that flu mRNA vaccines were being developed before COVID. Number one, to highlight the fact that there is actually a long history of working with these vaccines before the COVID pandemic. A narrative emerged that these vaccines were sort of rushed. They weren't. The COVID vaccines were based on years of experience with flu and other antigens. And I also like to highlight that because it's a good example of how basic research with one pathogen can kind of enable, in this case, a new vaccine against a SARS-CoV-2, which we didn't even know about when we were working with these studies earlier. So how can our studies of flu, help with the development of new vaccines? Clearly, the mRNA vaccine technology is very exciting and versatile. Whether some of the issues that we're talking about with what are the correlates of protection, well, that's going to differ from every virus that is out there. I really like this idea of multivalency and how far can we actually push it? 20 HAs is not the limit. There is a finite amount of vaccine that we can deliver. The doses of mRNA vaccine that are being used for coronavirus, that's probably, we're probably not going any higher than that. So the total dose is really limited, but the valency, what we actually include for mRNA molecules within that dose, is essentially unlimited. And I like to think beyond flu. Can we envision a multivalent vaccine that has 100 different mRNAs, including not only flu and coronavirus antigens, but antigens for many different viruses? Can we go up to 200? How about 500? I don't know what the limit is. So I think, and then you want to talk about regulatory on that front, then it gets really all over the place. But yeah, I think our studies hopefully will shed light on the development of vaccines for some of these other viruses as well.
Diane, may I interject here as well? Because
Yes,
I think
please.
there's also an answer from a different perspective, and that is that the mRNA technology has reduced the barriers to engaging in vaccine R&D across the globe. So I work at the mRNA hub in South Africa, and the WHO created this hub because the lack of equity in the supply of SARS-CoV-2 vaccines, right? And the intent there is to establish these mRNA R&D capabilities, starting with SARS-CoV-2 and create those local environments where R&D can be done on regionally important vaccines. So I think there is that perspective as well that goes beyond the thing, Scott, that you're talking about, which makes it very important, I think, as a technology. The other thing that I will say is, we may or may not recognize this, but mRNA technology is still in its infancy. There are huge improvements that are going to come over the next 5 years, whether it's room temperature stability or whether it's enhancing potency to get to the point that Scott just made. All these things are going to happen in the coming few years, and the potential applications are going to get much broader. So back to you, Diane.
Thank you. Yes, incredibly important points for sure. Okay, another question in the chat is, what is known about the durability of immunity provided by the multivalent mRNA influenza vaccine candidates? Are there durability studies ongoing, or are there plans for them, and how would you plan for them?
This is a great question. So what's the durability of SARS-CoV-2 mRNA vaccines is my question for the panel and the audience. And I would say against severe disease and death, it's at least 2 years for the most part. So we are looking at durability in non-human primates right now. We see very long-lived antibody responses, but we think those memory B cells and T cells probably stick around for a lot longer than antibody itself. If you look at memory B cells elicited by a flu infection, you can detect them 90 years after infection in humans. Famous study by James Crow at Vanderbilt. So we are looking at the durability, but I think it's likely long-lived, and we could probably learn from SARS-CoV-2 and the SARS-CoV-2 mRNA vaccines in this regard.
I would say what's long-lived, though, is probably the CMI. The antibodies we know are not long-lived. They crash very soon. But we know we're still protected, at least protected against disease and death. And it's the CMI that I believe that's giving us that durability.
So again, going back to the correlates of protection and defining those, and then having good ways to measure them, and then continue to measure them long term to look for that durability. So another question: what is the role of global regulatory harmonization in advancing access to next generation influenza vaccines, and how can it be facilitated? Maybe Amin, you could.
Well, I think that's a question for Norman.
Well, I'll start, but I think when you're dealing with things like pandemics, it has to be that there's got to be harmonization. I mean, we can't, and I think we did a fairly good job with COVID, with the International Coalition of Medicine's Regulatory Authority, to have those discussions between regulators. And really, if we lived the perfect world, we'd have these trials designed, 1 or 2 trials that we all agree upon, instead of all of these directions and all of by the regulatory authorities, and having a really harmonized acceptance of the clinical trials that need to be done to demonstrate the safety and effectiveness of the vaccine. That would go a long way.
And the only other thing I would say to that is, I hope we don't forget the lessons of COVID.
Yes, agree. Incredibly important to keep that in mind as we move forward. So we're at time. Unfortunately, it went by so fast. It was really fantastic speaking to you all. Thank you so much to our wonderful panel of experts today, and to the Sabin Vaccine Institute for hosting this event. It is incredibly important to continue to discuss and clarify these pathways to move towards optimization and use of universal influenza vaccines. And so I will hand it back over to Marissa for closing remarks.
A huge thank you to our panelists, and of course to Diane for leading this really rich discussion. Big thank you all around. And thank you to all those who tuned in. We will be posting the recording of this session on YouTube, and also releasing it as a podcast for those who prefer that format. And lastly, if you follow us on Twitter at The Influencers, you'll get notified of our next episode and of all the other work that we are engaging in. So thank you all again, and we hope you'll join us for the next episode.
Thank you.
Okay,
Bye-bye.
thank you.